Jai Hind everyone, I am Pankaj Kumar. Today's lecture is about what? The nucleic acid. In today's lecture, we will cover the following things. The DNA structure we are going to see, the triple helix or G tetraplex and RNA we are going to cover. So Chargaff rule, by the help of the Chargaff rule in the X-ray diffraction, Watson and Crick uh, give the model for the DNA. First nuclein was discovered. Nuclein is a acidic material and containing phosphorus that were discovered by the Frederick Misser. He was not knowing what it is, but they give the name as a nuclein. Later we found the DNA is that nuclein is the DNA, and it was discovered by the consecutive experiment of the Griffith and MacLeod, McCarthy and Avery, and later it is confirmed by the Martha and Chase experiment. So, Chargaff rule is what? Chargaff rule is telling you the purine and pyrimidine ratio will be always equal to 1. Or you can say the purine and pyrimidine sum will be always equal. Second thing, the adenine and thymine will bind with the 2 hydrogen bonds, and guanine will bind always with the cytosine with 3 hydrogen bonds. In case of the RNA, sometime single stranded RNA can form a double stranded structure. So in that case, adenine will bind with the uracil, not with the thymine. So if you want to check your understanding about the Chargaff rule, you can solve this question and tell me the answer in our chat box. There will reply the answer of this question. So X-ray diffraction and its interpretation. The X-ray diffraction looks like this. What do you understand by seeing this diagram? It looks like something X is there surrounding magnet. Something we cannot illustrate exactly. But if you will be have a concept about this and understanding about this X-ray crystallography, you can say it is like a some surrounding repetition will be there. Okay, something. It is a helix structure. So James Watson and the Crick used this two information, the Chargaff rules and the uh, Rosalind Franklin and the Morris Wilkins did this X-ray diffraction, and then they found this type of diagram. So the result of the Rosalind Franklin, Morris Wilkin, and the Chargaff, James Watson and Crick finally discovered discovered the model of the DNA. What they found, the DNA is a double helix structure, and these are anti-parallel strands will be there. The main backbone is made up of the ribose and phosphorus, and the this hydrogen bonds is between the two purine and pyrimidine. So they found there is a pitch which is repeating from here to here. You can see there is a pitch which its length is around 36 angstrom, and each pitch contain 10 nucleotides. In case of aqueous solution, the nucleotides number little vary. It is equal to 10.5. So two nitrogenous bases having a distance between the two nitrogenous bases will be 3.4. So it will be almost the number it is little bit not relating because it should be 34 angstrom. Okay, so it's because of the this structure. So secondary structure pitch length will be 34 angstrom, but this is the single second primary structure which is 36 angstrom. So it is because of secondary and primary structure. So the width of the DNA is around 20 angstrom, and there will be major group as you can see here, and there will be minor group between two. Major and minor group will help in the binding with the proteins like any hormones or any other growth factors like this. You can see this DNA will have the either it will be in the syn or anti form. So what is the syn form? You can see the differences in the arrangement of this nitrogenous base. So adenosine, if it is in nitrogenous base, adenosine is in the inside like this, then it is called syn adenosine, and if it is outside like this, then it is called anti adenosine. So purine will be can in the form of in the syn or anti, but pyrimidine will be always in the anti because it is a single ring structure. So, what is the cause of this? Because the steric constraint in the purine nucleotide are restricted to two stable conformation because of steric constraint. But pyrimidine, if you'll see, these are in the anti form only, and the sugar and the carbonyl oxygen at the two of the pyrimidine will be interference in the steric, like perform a steric interference, even though it will be only in the anti form. These are the B, A, and Z DNA. You can see B DNA is a right-handed helix, okay, and Z DNA is the zigzag form. You can see, and A form is the so will be right-handed. A form, B form. You can see the structure, and Z form. Z form is a zigzag and left-handed. Uh, the diameter will be around 26, 20, and 18 nano angstrom in A, B, and Z respectively. So helical turn will contain the 11 base pair in A, 10.5 in the B form, and 12 in Z form. The helix rise per base is the distance between the two bases will be 2.6 in A, 3.4 in the B, and 3.7 in the per helix turn. The angle between the two helix will be this much, and what it is? It will be vary in the A 20 degree, 6 degree in B, and Z form have a 7 degree. The C3 Indo-puckering will be occur in the A form, C2 Indo-puckering will be occur in the B form, and C2 prime Indo for the pyrimidine and C3 for the purine in the Z form. So this puckering we already seen. And what is the puckering? If like I can show you. Suppose there is a three fingers. Okay, suppose these three fingers are in the one plane. Now there will be because of some strain, because this cannot bind together each, so it will go upside, then it will called puckering. Okay, or it will go downside, so it will called indo puckering. It is going upside, exo puckering. So this is indo puckering of this third carbon, indo puckering of this two carbon, and like this. So A form is a anti glycosyl bond conformation will be anti form, and Z form will be anti pyrimidine and syn for the purine. These are the important thing you have to remember these things because of the palindromic sequence. You can see palindromic sequence will be there, and the palindromic sequence will cause the formation of either hairpin like structure or cruciform. So hairpin or cruciform will be looks like this only. If it is in the RNA, definitely it will form a hairpin like structure. In DNA, sometime it will form like a cruciform. So why this is happening? Because sometime if more adenine will become in a sequence, then that adenine will turns the 18-degree bending of the DNA. If 6 adenine will come, or like that, if more guanine will come, then it will also form a bending of a DNA. Why it is happening? Because if one protein is very far away, so the DNA will try to bend and go nearby that protein, like polymerase protein. So it will be help in the bending. That type of bending you already seen in the lac operon, like CAP and CRP protein was there. Okay, so in that is bending is happening. So what is palindrome? If you are seeing from the from back to forth or forth to back, if you will go read this, it will sound same. Like if you see rotor from first letter to last letter, it will called as a rotor, or from last to first, it will called as a rotor only. Now let's see the triple helix and G quadruplex, or you can see it as a G tetraplex. Okay, guanine. So you can see this is a thymine. This black is a thymine, and it is bound with what? Adenine. Adenine, if it is a cationic, then it will form another binding with the thymine. So there is a three bonds. Okay, so you can see three strands are there, in which two thymines are there. Means two pyrimidine strand will be there, and one purine ring is there. So this is forming a triplex structure. Guanine tetraplex is forming here, and you can see it is a 3D model guanine tetraplex. So the position where the this type of structure is forming, triplex structure is forming, that is called Hoogsteen position, and that pairing is called as a Hoogsteen pairing. So the reason is what? Because these are become protonated or protonide. Now mRNA and tRNA we are going to read. mRNA is what? Jacob and Monod illustrate the gene expression, and they propose the mRNA term. mRNA can be monocystronic or polycystronic. Monocystronic mostly in the eukaryote and polycystronic in the prokaryote, because the in case of the prokaryote, the single gene can give so many proteins. Okay, but in case of mRNA, monocystronic, one gene will give you the one protein. It is happening in the eukaryote. So this is called monocystronic. So mRNA is a single right-handed structure, and in the transcription we'll read more depth into the mRNA. So this structure you can see here. I will try to show. So here adenine have a different type of bindings. Okay, so adenine bind with the uracil, and this type of structure is forming, and 7-methyl guanine will bind with the guanine. There are different form of the nitrogenous bases again, or you can see here also adenine bind with N2 dimethyl guanine. It is not usually happening, but it is binding here, adenine with the N2 dimethyl guanine. Or you can see 7-methyl guanine bind with the guanine. So these all structure different, like unexpected structures or bindings you can see in the tRNA. So this tRNA usually is a clover leaf like structure, or in 2D structure, and 3D structure it is a L-shaped. The tRNA was first discovered by the Jamnick and Hogland, and one more scientist there, that is Stephenson. So this unusual base pairing you seen in the tRNA, this is a phenylalanine tRNA, and the first tRNA which is coded and discovered, that is a alanine tRNA. It was discovered by the Hole. So this is all about today's class. If you like these videos, please like, share, and subscribe. And please go and solve the questions related to this topics questions, which is given in the description box. If it is not there, you go and download the uBiology app. It will be present there, and keep asking the questions, and so I will be very active, and I will update the questions and videos. Thanks for watching. Jai Hind. We'll meet in the next video.